on time-ish, but not according to that clock. It's my true pleasure to introduce one of my colleagues and our last speaker of this workshop. So Paul François is a professor of bioinformatics and biophysics at the University of Montreal. He obtained his PhD in theoretical physics from the École Normale Supérieure Université de Paris 7 before completing a postdoc at Rockefeller University. His group's interested in biological physics using theoretical and computational modeling in collaboration with experimental biology. His long-term research goal is to build predictive theories for biology, and he and his team are working in close collaboration with biologists, building models of complex biological systems from the subcellular to evolutionary scale. So thanks, Paul, for finishing up the workshop. We look forward to it.
Thank you so much for the invitation. Yeah, I'm sorry I could not be here earlier during the week. I had a bit of a crazy week, but I'm very happy to be here today and I guess to finish the workshop. So I will tell you about some work that has been going on for quite a long time, actually. So something that started about 10 years ago, and that is leading us to now actual clinical application. And as you can see from my title, it's related to T-cell recognition. And so I decided to structure my talk in a semi-historical way, like especially since most of you are mathematicians or theoreticians, starting with the theory and then moving towards application and experimental data. And so I will mention my collaborators later, but this has been done in close collaboration with experimentalists, in particular Grégoire Altambonnet at the National Cancer Institute at the NIH in Bethesda. Okay, so what's the problem? So I guess you're familiar now with, I mean, you're here to study oncology, and I guess you've heard about many times already about the immune system. So my work focuses on the adaptive immune system, and we're going to start with a T cell. And so as you probably know, T cells are here. So this is my drawing from Wikipedia of the T cell. They interact with antigen presenting cells. And so the antigen presenting cells, what they do is that they digest the peptides around and they just present to the T cell. And then the T cell has to decide if this peptide is self or not self. So that's the problem we're going to start with. So a peptide is presented. I'm going to call that a ligand. The ligand is going to come from your body or from something else. So we're going to say self or not self. And then the T cell interacts with it through its receptors. And so an important thing in T cells is that every T cell is different, but in one given T cells, we have identical receptors. And you have roughly 30,000 receptors per cell. So all the ligand interactions, receptor interactions, they're done with the same receptors for a given T cell. And so now you can have many ligands presented to a given T cell at the same time. And based on this, the T cell needs to take a decision about to trigger an immune response or not. So now the first question is that what is the biochemical difference between self and non-self? So that's an important question. And to some extent, it's not fully solved yet, but we're going to assume following some ideas that have been around for a long time and that were somehow checked experimentally as well, that the main determinant of what I will call the ligand quality later in my talk. So whenever I talk about quality, I will mean this parameter, tau, which is a typical binding time of a ligand to a receptor. And of course, this is what I mean by tau is average time. So of course, it's related to biochemical rate, and this ligand will typically dissociate with the rate of 1 over tau, okay, of equals 1 over tau. Okay, so what is the difference between self and non-self? It's relatively simple. So basically, if you have a long-lived ligand, a long-lived bound, it's not self. So your ligand stays there, it sticks, and so the cell might trigger an immune response. And then on the left, if the ligand dissociates very rapidly, it's self. And so you have a relatively sharp threshold. Where above, say, like roughly 4 or 5 seconds, those are real units. Below, you're essentially self, you don't trigger any immune response. Above, you're not self. And we'll revisit this later to comment on some interesting phenomena happening right in this region. Now, the puzzling thing, and that's really what drove me into this problem, is the following. So, of course, you have many self ligands in your body. And then a virus or whatever is going to want to hide from your immune system. So typically you don't have many not self ligands. And so now, so if you try to put actual numbers on it, and this coming from experiments as well. So typically, if you have like, essentially if you saturate the receptors with self ligands, you don't have any response. The T cell, because it should not be, because of course you have many self ligands in your body, so you don't want to trigger any response. But on this side of this axis, you want to be able to detect very few ligands. And in fact, T cells have really close to molecular sensitivities. And so that means on average, you would expect, something significant response around, I don't know, something like 10 ligands. I mean, this is kind of like estimates, but it's known that T cells can respond to a single ligand. This has been shown experimentally. So they are very close to, the best they can do. Okay, so now, so what really drove me to this problem is this kind of contrast between, you look at the concentration difference here versus the time here. So you see there's a kind of tension here between the two, because you don't vary much the binding time, but you can have a huge variation of the concentration. And why is there a tension? Is because you typically expect the response to be a function of both the concentration of the ligand and the binding time of the ligand. That's what you expect naively from a model. And in fact, so this really led people to study this problem more carefully. And in '95, McKittan proposed a relatively simple model based on KT proofreading. So KT proofreading has been proposed by Hopfield and Inyo, Hopfield Physics Nobel Prize 2 weeks ago, so KT proofreading to actually understand how DNA was replicated and how the mutation rates of DNA was far lower than what was expected from thermodynamics. And then Hopfield proposed this KT proofreading concept that was adapted in the context of immune recognition by McKittan. And so to make a long story short, and I'm going to cheat a bit because if you do the actual math, it's not exactly the result I'm going to show you, but for the sake of time, I would make simplifying assumptions to explain why there is, how it works and why there will be an issue later. So what happens is that you have ligand receptor interactions. So that's kind of standard biochemistry, K on, and then you have a binding time. And then what you assume is that the receptor, once it's bound to the ligand, can essentially go through steps, change states, and then you have several steps corresponding, for instance, to phosphorylation, phosphate group added or something like that. So actually, in the actual immune system, in the actual receptors, we know there are phosphorylations that are occurring. So this is something that is biochemically realistic. And so we're going to add phosphorylation, phosphate group to the receptor. And the important thing that is happening, so if you write the biochemistry of this, is the following. So the ligand can dissociate with the same rate, 1 over tau. But then whenever the ligand dissociate, what is happening is that essentially all these phosphate groups, they disappear. You essentially reset the receptor to its normal state. So that's KT proofreading, because it means that, if you need to wait long enough to reach the last complex, because then the minute the ligand dissociate earlier, boom, you go back here, and then you have to start again from the beginning. So that's why this is called kinetic proofreading, because there's a kinetic effect. And our field showed that you needed to inject energy to do that. I mean, there are very interesting, cool physics going on in KT proofreading. But in the context of immune system, what that means is that you can compute on average the number of fully phosphorylated complex, like the number of fully engaged receptors, essentially. And so you can do the math. And then again, simplifying assumption, roughly what you see is that the total number of fully phosphorylated complex is going to be roughly proportional to the concentration of ligands. So I'm going to call that L, the quantity, and to an exponential factor, to a geometric factor to the power n, where n is the number of steps, and it's roughly equal to the binding time, tau. So that's the first important result. KT proofreading gives you something where proportional to the concentration, and then a function of the binding time to the power n. So that's important. Okay. Okay. Fine. So that makes sense. You expect that the more signal there is, the more fully phosphorylated complex there are, and you expect it to depend on the binding time. And the only thing that is a bit unusual is this power n, but that is due to these n steps. Okay, so now let's see in the context of what I've just told you what that means. Okay, so to be a bit slightly more quantitative and slightly more realistic of what can happen in biochemically. So let's take the log of CN, and let's say, so of course, this is just the sum of this log. And let's assume that CN is triggering a response. Oh, yes, sorry.
Yeah, hello, I'm sorry to interrupt.
Oh, please do.
First of all, thank you for the amazing introduction. And my question is, well, it's known that T cells, that they generate this selective part of T cell receptors just by like random processes, which are therefore selected.
Yeah.
So what stops T cell from producing like this part, which would be, which would match the self antibodies?
Oh, I see. So there is this process called thymic selection in the thymus, where you basically make sure that you exclude the T cells that will kill yourself. So it's actually a relatively complicated process. You know, you have what's called negative selection, positive selection, and you know, my understanding talking with immunologists is that there are some models around, but it's not fully understood, and there is some debate about how it works. But basically, what makes sense is that we know there's a selection process that prevents your immune cells to kill yourself, basically. Okay. So coming back to my kinetic or McKesson kinetic proofreading model. So let's assume that you trigger a response when CN is higher than some concentration. So let's go into a log-log plot where we plot the log of quantity L and the log of quality. Remember, quality is I mean tau, and then of course this is just a straight line, okay? And this straight line, above the straight line, you know, this defines the threshold of activation. Above the straight line, the system is on, the T cell is triggered. Below, it's off, okay? And so this straight line makes tons of sense. You know, it means that, okay, let's say I'm just above here, I'm barely triggering response. Well, that means that in this model, I could, if I'm here where I barely trigger response, I could have an equivalent response by just lowering the concentration, but increasing the quality, which makes sense. Again, you know, you expect to have a kind of trade-off between concentrations and qualities. That makes total sense. Okay. So now let's put back what I told you. So we know there is no response here for 10 to the power 5 ligands, and then we have a response here for roughly 10 ligands, and then I put the binding time, say 20 here, and here, like say 2, 3. So that means that I have, you know, my boundary should, you know, look like something like this. We need, you know, I don't know what the exact shape is, but the point I want to make here is that, especially if those guys are really up there, you know, because there is no response. So it's kind of a lower boundary. So you basically need something almost vertical. And that is really the mystery. So something vertical here, if you look at what that means, it means that you're only sensitive to the binding time. You basically don't see the concentration. So that's a bit of the mystery. And then, okay, so here I'm undoing a little bit. I told you the actual model is not exactly like this, but you can really make this very rigorous. You can also look at how fast the response will be, because kinetic proofreading has a big issue. It's known that to be efficient, it needs to be slow. So if you have a T cell, you know, we know the T cell has to respond within a couple of minutes. So that means that it puts constraints on the kinetic proofreading. So in the end, you can do a calculation, and you can show that to have something realistic in terms of biology, you need n equals 20, which means basically, you know, many phosphorylation steps. That makes no sense. But still, you need to keep this molecular sensitivity. So that is really what drove me into this problem, because there was something very mysterious on the biophysics side. This model cannot really work. And so we had to look at other ways to do this. Okay, so what's the way to do this? Well, actually, it turns out that my friend and collaborator, Grégoire Altambonnet, published a very interesting model in 2005 that was very much, you know, system biology-like model. You know, he really tried to integrate everything that was known about the system. And so, you know, there are things that are known, you know, all kind of phosphorylation. You know, it was kind of like the golden year of systems biology. So you try to put everything in a big model. But this model works remarkably well to explain what is happening. And they could test many things in this model, many non-trivial phenotypes that I will tell you about later today. That works. This model works very well. And from a biophysics standpoint, for me, I mean, when I saw this model, I really could not understand at all what was happening. And so what drove me to this system was really to follow, you know, Gunawardena quote that I like very much. You know, is there a way to distinguish the biological root from the molecular tree? Like trying to simplify this model against an intuition about what is happening in this model. That was really my primary motivation to study this problem. And so, well, we worked on it. And so what we found very interesting results. So we tried to first simplify this model, and we also did something which I've been doing for years, which is to try to find a solution of this problem of very, maybe I'm going to draw this because that's going to be very relevant later. I want this. I want something that is essentially on this right, on the right, and off on the left. So this problem, actually, you can formalize it. And I was doing, especially that's something I developed a long time ago during my PhD. I've been developing algorithm to basically build network doing stuff, and I give that to my algorithm, and I ask my algorithm, okay, build a biochemical network that is able to do that. And so that's a program called that we know called FIVO. Okay, it's doing this. And I give that to my program, and that works. And to my big surprise, it gives me an extremely simple network. So that was my first thing. And this is literally this network, 3 equations, very simple. So you can solve them. If you have 5 minutes to solve it or 2 minutes to solve it, it's very easy to solve. You see what happens. And I explain to you what this model does. I mean, the equation does not really matter. I'm going to give you an intuition, a very, very, very simple intuition about what is happening here. Okay, so what is happening? Okay, so now how can this work? So remember, I want to do this. I want to have something that is essentially insensitive to ligand concentration. So that sounds complicated because I just told you, you typically expect the things to be sensitive to both concentration and both binding time. But one thing I know I can do something like KT proofreading that will give me a line like this, a slope. Okay, so now then I told you, well, a simple way to get a vertical line is to get n to infinity. N to infinity gives you a vertical line, but I told you that's not possible. Basically, you don't like infinity in biology. But now imagine the following thing. Imagine you can compute. You have 2 biochemical networks or the same biochemical network combined in some way that is doing 2 lines like this. So maybe one part of the network is going to do L tau, and one part of the network is going to do L tau square. And that's very easy to do because I have a cascade. Now, if I combine those 2 things, and let's say, for instance, make a ratio of those 2 part of the network, then I have something that is only sensitive to the binding time. And then if I have something that is only sensitive to the binding time, well, that means I can do essentially a detection irrespective of the quantity. So now I'm going to show you that I'm going to explain you how this simple model I showed you is working. So usually I do it on the slide, but since I have a blackboard, I'm going to use it. So that will be easier. Okay, so how does that work? You get your KT proofreading cascade. Okay, and so what you expect from KT proofreading, I told you like Cn will be roughly something like L tau to the power n. That's the output. And then maybe I have all complex in the cascade. So let's say Cm here is going to be proportional to L tau to the power m. Again, it's not exactly this, but by and large, it works. Oh my God. Sorry, okay, so how is the system working? And we call that adaptive KT proofreading. So now let's see. So for instance, this guy here, what he can do, he can activate something, let's call it S, a phosphatase or something. And then it's very simply what is happening in that S then, what you can do, it can maybe repress the last step in this cascade. So now if you do that, S, you expect it to be proportional to L tau to the power m. And then when you combine, so now it represses this. So that means that instead of having something like this proportional to L to the power m, you are going to modulate the rate in front of this term, and that is going to be modulated in a negative way. So for instance, you get something more like 1 over L tau to the power m. Okay, and then if you do if you look at this expression, then the L simplify, and you get tau to the power n minus n. Okay, so that's the way this adaptive KT proofreading working. And the reason why so the KT proofreading part is clear. It really comes from this cascade. And the reason why it's adaptive, it really looks like classical biochemical adaptation from the 2000s when we were doing system biology and everything. You do a feed-forward negative interaction that is getting rid of the concentration terms to get something now that depends purely on the binding time. Okay, and so that's the way these things work. So there are several consequences to that. One consequence that I come back later on this is that this negative feedback loop. So now you have to think that those are several receptors. You know, you have several receptors at the surface of the cell, and this CM, this CN are the average number of receptors for a given cell. And so when you activate something here, you're activating it inside the cell that is shared between receptors. So that means that effectively the receptors talk to one another through this species, and this species act negatively. So that means that the receptors are negatively coupled. That's something that will come back later. Just from, but you know, I mentioned this now because we will need that later. Okay, you're still with me? Okay, if you understand that, everything is easy. Okay, so I'm going to. Okay, good. So I'm going to move forward a bit in time. So we proposed this in 2013, and at that time we had some experimental evidence of this. And I will comment on some of them, but at this stage, I think it's better to fast forward in time and show you one thing we've done very recently that is really consistent with the model, and then we'll come back to things we've done before, and then we put everything together. Okay, so the model I told you about is really a model at the single cell level. So it's really one single cell, but then of course you have many T cells, and also that's a model that will work within a couple of minutes. But then the T cells get activated, and then you trigger the immune response. You have a cytokine secretion and things like that, and then you get sick. So for instance, as you know, for instance, something like IL-2 is secreted, and then you get for a couple of days you get fever. So there's a lot of dynamics happening here downstream the T cell, and so. And then cytokine dynamics are quite interesting because also we know that things can get wrong and you can be killed by your cytokine dynamics, basically. So that's an interesting problem by itself. And so with my colleague and friend Grégoire Tambonnet, we got interested for various reasons, including this little model, but many other reasons as well. We got interested into cytokine dynamics. And so then you need to study cytokine dynamics. And so what Grégoire did was really clever and really interesting is that he built a robot that was allowing to study cytokine dynamics very systematically and in a very quantitative way. So this robot, what it does is that you have 96 wells, okay? And these 96 wells, you put OT1 T cells at different quantities, and then you present them with antigen. You stimulate them with antigen-presenting cells, and these antigen-presenting cells have different peptide, different quantity of peptide, different quality of peptide. And so you put that in test tubes, and so now you have 96 little immune response in test tube, and then you just conduct time series regularly, and you measure everything you can. So we started very small with a couple of cytokines. Now we have many more cytokines, many more markers. But what I will tell you about today, this is really the early days. We only had a few cytokines, but you can do a lot already with very few cytokines. Okay, so you run those experiments, and the data look like this. So I apologize. I realize it's not very readable. I will walk you through this. So those are the data. So each column here is a different antigen for the OT1 system. So you go from weak to strong here. Okay, so that's antigen. Each of the line here is a different cytokine, so you don't see the name. This is IL-2, this is interferon gamma. This I cannot even read on my screen. I can read it, but on the screen, this is well, whatever. IL-6, IL-17, TNF alpha. Okay. And so now each of these box here, the same time courses. So you don't see the units. This is about 70 hours. So some time course on cytokines. And the different colors are different conditions. So different T cells number, different concentration. So I don't want you to look carefully at every single line. My point here is we can generate those data and we can systematically vary the parameters and study what happens here. And so we can do that. It's very systematic. It works very well. And when you look at it, just from a bird's eye perspective, you feel there is a structure. Like there seems to be something. So the first question is that what is this something? Like how do you extract the structure? How do you understand the structure from those data? And so since we were interested in ligand quality and quantity, especially given our adaptive connective proofreading model, we expect the output of the system to be pure quality. So that's a question we ask. Is it pure quality? Can you quantify it? Could you identify modes that are not pure quality? And then what you do those days, and I think that's reasonable, especially when you have data like this, you need to do a bit of machine learning to understand what is happening. So that's what we did. We did a bit of machine learning, and I'm not going to dive too much into the details of machine learning because that's not very interesting. What is interesting is the result of the small neural network approach that we did. And the important thing that, so to see if that was, if it was understanding quality, we tried to build a classifier, taking the cytokine time course and essentially giving you the name of the antigen that was presented. Okay, so that's what we did. And the important thing that we did because we wanted to have a sense of what was happening is that we put in the neural network, we put a bottleneck. So it's very similar to autoencoders in machine learning, if you know autoencoders. So we put a little bottleneck with only 2 nodes. And the hope is that, by doing this, essentially you kind of project your data in 2D, and then you can analyze them in 2D and see what happens. So that's what we did. And so the details do not matter, but now we're going to focus on this. And then I'm going to show you how the data look in this 2D latent space that was derived through training 4 categories of the antigen. And so those are my data now in 2D. So those are my data in 2D. And so I was trained to detect antigen quality. And so you can see why the neural network is working very well, because you see that these different colors correspond to different ligand quality, different binding times. You see that each corner of this latent space, you have different colors. So that means that you can understand why you can classify it very well. So that works very well. You have the quantity, the number of cells, and you see they kind of like all together. Okay, so at least our neural network, we know from that, we know that we can detect, or the system can detect ligand quality based on the cytokine time course. Okay. But then the interesting thing is the following. We realize that actually this is not only one representation of the data, but this is a complete representation of the data. And so the way we did it, we actually trained a small decoder, and then we showed that from this latent space description, you can actually completely reconstruct the initial time courses. And so that means that this description of the data, these trajectories in those latent space coordinates, and we can reconstruct what this is, but you don't need to know what it is. It's a complete representation of data. It contains all information you add in the cytokine time course in the beginning. Everything is contained here. So that's a dream for people like us, because that means that we can focus there, understand everything what is happening there, and you can then go back to the full system and understand the data. So that works very well. And the thing that also what actually means is that if you have trajectories here that are different, unless you have a very complicated decoder, but if the trajectories are different, you expect them to be also different in the data. So that means that if you don't have overlap here, it means those are different trajectories. And so in particular, if you see here that you have very nice quality differences in this latent space, it means that in the full data set as well, it's mostly quality that is determining what the patterns in what we saw. So that means already we see something, we see that it's only mostly quality driven and not quantity driven. We already see that from that. So we are very happy to see that. Frankly, we are not expecting that. Like we thought we would get much more concentration dependency, and we looked hard for concentration dependency, and there are a bit of concentration dependency. I'm not telling you there is no concentration dependency, but we were hoping to have something less clear. Well, data are data. But we're happy because that was consistent with what we were saying. Okay, so to prove a little bit my point. Okay, so now it's actually interesting. So I told you that these things discriminate based on quantity, but to be a bit more rigorous, you could still have things like different behaviors in these trajectories. So what we did, we tried to fit those trajectories, and so this is kind of like very, very stupid trajectories because you have time as well. So I'm a physicist, so a physicist, I think about cannonballs and stuff like that. So we use a ballistic model to model this. And so we can define a set of parameters to define those trajectories. So we have like the speed, we have the angle, and then we have this kind of thing where it goes down. So we define a parameter T0. And then what we saw when we fitted those curves, we saw that those parameters are highly correlated. This is what you see here. And not only they are highly correlated, they are highly ordered. So you really follow, like you see, got V4, T4, Q4, and 4. And this order here corresponds to what is known about the antigenic strength of those various ligands. So not only do you learn that there's only one parameter controlling the system, you learn that it's continuous, and then the system it's continuous, and remember it was trained only on classification. So it means in our latent space description, we have some kind of hierarchical variable, hierarchical parameter that is really spontaneously built in our latent space. And so that means that you can actually literally compute, the position on these curves gives you the antigenic strength. You can really define this as the antigenic strength because this is what defines the cytokine time course. So you can really take a ligand and then define antigenic strength based on this behavior. And so we can call that antigenicity. And this is a continuous parameter. So we have a very well-defined notion of antigenicity that is coming from this description. Yes?
Hi, sorry to interrupt.
No problem.
Could you please explain one more time about the latent space and what is LS1 versus LS2?
Okay. So think about first principal component analysis. So take your data, usually you do PC1, PC2, and that could be a way to describe the data. Okay, so now instead of doing this, we're doing something slightly different. We're taking a neural network, and we're taking a neural network that we classify the antigen. Okay, and so if you were to take a purely linear neural network, what we do would be very close to PCA and looking at the first principal components of the first two principal components of the system. This is well known, especially if you do an autoencoder, linear autoencoder, we know it makes principal component analysis. So instead of doing this, we put some very small nonlinearities. And so the small nonlinearities will be able to essentially capture the nonlinear variation in your data. So that's essentially what we do. It's like a PCA that is slightly nonlinear, and then we're looking at the first two components, and because it's slightly nonlinear, we can actually make it capture every variable, every variance in the data with only two variables.
Okay. The second question I had was in the data that you were feeding into this.
Yeah.
And you were adding different cytokines. Did you add the combinations as well, or just individual cytokines?
Oh, we give all the cytokines at the same time.
Oh.
Yeah, so we really listen to the neural network. We literally give, like here, you give like 7 cytokines. Actually, I did not show them all of them because only 5 really matters. But we give the 5 cytokine time course at the same time.
All onto the same.
All at the same time. And so those latent space variable actually, well, I don't have a slide on it, but we can actually reconstruct how they depend on the cytokine. So for instance, LS1, actually you see it here. LS1 is highly controlled by IL-2, but it's also controlled by the other cytokine. While LS2, it's mostly IL-17, and also like you see IL-6 TNF-alpha that is controlling LS2.
So when you said you're changing the concentration, you give like all 5.
Yeah.
You change the concentration of one of the 5?
No. You mean in the no. The setup is that we give, so let's say one experiment, we give a quantity of antigen and a quality of antigen. So we give some antigen, okay. And then we monitor the time course of the cytokines in each single experiment. So each line here, it's one experiment. Okay, and so now what we give here is for each experiment, we give the 5 cytokines as a function of time. So we use the 5 cytokines. We give one time point for each cytokine. And then we use all the data, all the time points to train the network. Yes? No? Okay. No, you okay, maybe we can go on offline. This is really simple. There's really nothing mysterious. Like we just give the data and then Oh, the experiment. Okay, I can go back on the experiment. No. You get all the cytokines at the same time. In one experiment, you get all cytokines at the same time. So you have a 5-dimensional vector characterizing your test tube.
But when you give the experiment, you're giving cytokines plus antigen?
No, we're just giving antigen. This is endogenous. Yeah. We give the antigen, and the cytokines are secreted. So this is really like we're monitoring the immune response. We're not adding cytokine. We're just looking at the response in test tube.
I'm a little bit confused. Like in general, this type of cytokines, they have like different rates of activation. So in time, like sometimes some are quicker to like be produced, but like how does this? How can that be accounted for that?
But that just means that just means they're very, even though I agree with you, they could have different time of activation, but we can still capture them because what that means is that, for instance, let's say you have two different time scales for two cytokines, but then if you put a stronger antigen, if they're moving in the same way, even though they have different time scales, they're moving in the same way, it means that they're all related to one parameter. This is kind of what is happening here.
Thank you. It's a beautiful way of using neural network to do my mechanistic insight. I was just wondering, so you're giving the full time series.
Yeah.
So how do you make sure that in your latent space, you're maintaining the same idea of time? So
Okay.
like, it's probably very technical.
This is really going into the little tricks we use to do that. But okay, so what we realized, so first, there is one step here that I did not tell you about because I don't know if it's interesting or not, but okay, since you asked me the question, if you give only the time points, it's not really working. Like if you give single times, it's not really working. What we did is that it's much better to just do the time integral. And so we give the time integrated time courses. And for all kinds of reasons, it's much better. It's much smoother. It's much more robust. Like it really helps a lot to do this classification. So basically, you have memories. So so because of that, the trajectories are correct. So this is what you see here, and you already see here that they are very, very, very. They're already nicely layered. So right at this stage, they're nice here. So the neural network has very little job to do to separate them. Okay. Yes.
So just to clarify that I understood the experiment. So you give antigen and you are measuring the
Yeah,
five
we are
cytokines.
measuring the cytokines.
Okay, so I wasn't that you were giving the cytokines.
No. We give the antigen. Sorry about that. We give the antigen. We're measuring the cytokines, and now we're measuring markers as well. So I will not show you the markers, but you generate here. It's a 7-dimensional space, but now we have like 33-dimensional space. So and anyway, okay, good. Okay. So the take-home message of this is that we can infer from the cytokine time courses antigenicity, and then what we can do. So that was trained on some mice data like OT1 mice. But what we can also do is that we can test them on other mice, or we can even test them on human T cells. And so now what I show here is this parameter, this antigenicity we measure in our latent space. So let's call it V0. And it turns out you have other measures of antigenicity, like the EC50 of this antigen. And so you see what works quite well because you have really a perfect correlation between the V0 and the EC50. And what is really remarkable is that we train them on OT1 T cells, but it really transfers not only on other ligands from OT1. So for instance, the Y3 and A2 were not used in the training set, but you still have a very nice correlation between V0 and EC50. But it really transfers very nicely, without any change, it transfers to other type of T cells. And also it transfers very well to humans. So, we train, so what that means that quantitatively, the cytokine time course are very, very similar in all these different contexts, is what we find. Okay, so that's why we say this is universal. Okay, so now the main point of this, one of the main points of this that we see that the response at the cytokine level is essentially a function of antigenicity, and there are a little bit of influences of other parameters, but this thing is able to essentially detect binding time and is a very, very essentially nice function of binding time. Okay, so now I'm going to take a step back because we know something by our study of adaptive kinetic proofreading that is very characteristic of this kind of systems, which is ligand antagonism. So I saw there are some posters on ligand antagonism, so I don't know if it's something you've heard about, but I'll just explain in a nutshell what ligand antagonism is using this adaptive kinetic proofreading model that is still on the blackboard. So let's say you have a ligand just above the threshold, say 5 seconds ligand, that is triggering response. Okay, so this is when you present one ligand, we have a response, and everything I've told you so far, we're only presenting at the same time only one type of ligand. But of course, a T cell has multiple receptors, and you can present different receptors at the same time. So let's present at the same time ligands that are just below the threshold, so 3 and 2 seconds. So ligand antagonism, what is happening is that if you present ligands below the threshold, you can sometimes essentially cancel out the response. Okay, so it's really easy to understand what happens looking at a model like this, and so I'm going to do it in one slide. Why do you have antagonism? Okay, So let's say, so now we have to go from one ligand to two ligands. So let's assume I have two types of ligands. I'm going to say the quality or the binding time is tau1, and I'm going to say assume tau1 is higher than tau2. Okay, so in a classical kinetic proofreading scheme, you know, if I forget about my S over there, I expect roughly if I have one type of ligand, say tau1, I will have you know, my CN would be roughly L1 tau1 square. Say if I only have two proofreading steps. And now if I add another type of ligand, assuming there is no competition between the receptors, which is what we have actually, we see antagonism without any competition, then you kind of adding up the influence of two ligands. So if you have a simple kinetic proofreading scheme, you essentially expect that the minute you add more ligands, whatever they are, you increase the response, you increase the probability. That's what you expect. That makes sense because, you know, this is quite linear in terms of concentration. But now if I do this, you know, if I have my adaptive kinetic proofreading, I'm doing a very different computation because now I activate a negative thing, and this negative thing is going to change my output. And so I'm going to do this ratio now. And it's not very difficult to see if I do the ratio, you basically revert this. And the intuitive is, I mean, you can do the math, it's very easy to show, but the intuitive reason is that this thing is more like a weighted average of tau1 and tau2, while this thing is exactly equals tau1. So if you do the weighted average of tau1 and tau2 is lower than tau1, well, you necessarily have this that is lower than this thing on the left. Okay, so that's antagonism because you add tau2 to the mix and you lower the response. So that's very easy to understand. So my explanation, when I give this explanation to biologists, I say, you know, what you have is that you have negatively coupled receptors. And so the kind of the low ligand quality, they kind of activate a bit more the negative feedback. And then there's a balance happening, and you can essentially kill the output of the signal. So that's what, you know, this is a very complicated sentence to just explain something like this happening. Okay, so that's interesting because we expect, you know, like we know that in T cells, we expect antagonism. And actually, this is something we proved a long time ago. We see that in T cells. I was a smoking gun for this kind of adaptive kinetic proofreading model. In fact, I even have a small theorem. I can show that with some conditions, if I have a discrimination that is, you know, purely depending on tau, I can show that you necessarily have antagonism. It's really easy to do. What you do is that you put yourself at the boundary, you tailor, expand what happens here, and you can really show that you expect antagonism to happen. It's very easy. Okay, good. So we expect antagonism. And so now I've told you on the cytokine, we seem to have something that depends on, you know, the binding time. So according to the theorem, you should have antagonism. And fortunately, we do have antagonism. So here now I'm showing you, so I just pick one cytokine, IL-2, okay? And so we're looking at IL-2 as a function of time in our robot, okay? And so let's stimulate our system with N4 and 10 picomole, so very small concentration of N4. N4 is a strong agonist. You see, you know, IL-2 going up and down, boom, okay? So that's what you expect. You have an immune response that is you know, quite actually relatively strong because N4 is very strong. Even at 10 picomolar, you get a response. But then when we stimulate the system with 10 picomolar of N4, and at the same time, we put 1 micromolar of V4. V4 is a ligand with a tau2 that is much smaller. You see that basically, I mean, you're not following N4, you're following the V4 thing. Okay? So that's antagonism. Because normally what you would like to do, if you think in terms of immune system, you will still like to detect the N4. You know, N4 is probably a bad guy. So you would like to detect it even if you have another thing that is benign, you know, like V4 isn't doing nothing. So that's a bad thing, you know, like your immune cell is not able to detect N4 10 picomole when you have V4 around. So that's a bad thing. But we see that experimentally. And so this is kind of a smoking gun for this kind of negative feedback adaptive proofreading thing that I'm telling you about. Okay, so that's a bad thing. And this is where, okay, so now it's a bad thing, but that's a bad thing we can quantify. And so now I'm also, yeah, I think I have time to use the blackboard to explain to you. So we got really interested into quantifying this antagonism thing. And to do that, you know, experimentally, we decided to focus on something that we call antagonism fold change curve, which is a very complicated name for something very, very simple. Okay, so to explain what I mean, okay, we're going to draw a graph. Okay, where I'm going to call tau1, this is the binding time of the agonist. And this is fixed. This is my N4. And then tau 2 is the binding time for the antagonist. And so what we want to do is that we want to plot as a function of tau 2 the strength of antagonist. And so that's very easy, because what we can do, we can use this kind of model with the ratio that I told you about. So for instance, I'm going to illustrate it on the model, but then of course we're going to do experiments to measure it. So in the model I told you about, at a simple adaptive KT proofing, if I have tau1 alone, you know the response is L1 tau1 to the power n over L1 tau1 to the power m. So this is tau1 to the power n minus m. So that's easy. Now if I have tau1 plus tau 2, okay, well, I have something like L1 tau1 to the power n plus L2, sorry, tau 2 to the power n divided by L1 tau1 to the power m plus L2 tau 2 to the power n. Okay, so now what we're going to do, we're going to compare those two values as we vary tau 2. So that's what I'm going to draw here. And so to compare the two values, I'm just going to make the ratio. So I'm define my antagonism fold change curve. Antagonist fold change curve. Okay, this is simply going to be 1 over tau1 to the power n minus m times L1 tau1 to the power n plus L2 tau 2 to the power n divided by. Okay, so I'm going to look at this curve. I'm going to fix L1, L2, tau1, and the only thing I'm going to vary is tau 2. So let's see what happens. So for tau 2 equals 0, you forget about this. You get tau1 to the power n minus n divided by tau1 to the power n minus n, so you get 1. Okay? And so then I'm going to draw a line because I'm going to look. This is my reference level. When I only have agonist, I should expect this because this is what the response for, this is a 1, you know, a rescaled response. Now, let's look at what happens for as tau 2 vary. It's not too difficult to see what happens. So for tau 2 equals 0, you get here. Okay. For tau 2 equals tau 1, if tau 2 equals tau 1, you actually get again 1. Okay. So you have a curve that is going through here and through here. So then you can apply a Wall theorem to say that you're going on one direction or the other direction, initially at least. You can compute the derivative. It's not very difficult to see the derivative in 0 is negative. So you expect a curve like this. Okay. Yes.
There's a question in the chat. Sorry.
Yeah.
How do we know the dampened response isn't due to competition at the receptor level?
Okay. We know it. Great question. We know it from different lines of evidence. So first, if you look at the number, you get antagonism for very low concentration. So you cannot get competition. And first line of evidence. Second line of evidence, there is a classical experiments from the late 90s, actually early 2000s, where they could really show that if the antagonist are the other side of the agonist, you see the negative feedback signal via phosphatase Cauchy point traveling from one receptor to the other. So we know that this is not saturation. We know it's an active process leading to this negative thing. But yeah, and I show you what happens in CAR-T. These are different receptors. You get antagonism, so that's another evidence. Okay, so now antagonism is due to the fact that you're going down here. You know, whenever you go below 1, you have antagonism. So this is this antagonism for change curve. You know, this is just, I usually do it on slide, but I use the board, so that's better. That's antagonism. Okay, so we can measure that experimentally. And so we did it, okay? And so now we are kind of putting together everything we've done. So those are experiments from 2 years ago or last year, where we measured this antagonism fold curve change. So you don't see them here, but they're here. So this is a black thing here, and the red thing here are those antagonism fold. So they have kind of the right shape. They're going down and then up again. So you see the red thing. You see those are different concentration. The legend is hidden, but you see. But then what we realize is that the classical model that we had in 2013 is not working. And in particular, so we try to fit it. But what we see is that the model is somehow completely underestimating antagonism, which was a big surprise to us. Like our model that developed 10 years ago we knew there would be antagonism, but there are much fewer antagonism in our model that you observe actually. So we had to update our model to fit the data. And that works very well. And actually, the new model turns out to be simpler than the old model. We had a bit of trouble to find it, but basically, simplicity was key. Like we tried to do very complicated stuff. In the end, something relatively simple is working very well. And those are those antagonism curve, fold change curve, experimental one, measured in the robot. Okay, and you see they all go down and up. And so these different colors here are different antagonist concentration. So 1 micromole, you have more antagonism than 1 nanomole typically, except here. So what was really difficult is that they actually not go all through to one. Experimentally, the difficult thing is that experimentally it's not like this. If you vary the concentration, you start having a weak L2 effect. So you start having a motion like this. So this is like this is high L2, and this is low L2. Okay, so that means that this model, simple model, doesn't explain the data. So we had to tweak the model to make it work. But that works. So we can explain the data very well now. Okay, and just to give you a sense of the model, it's really simple. I mean, like it might not look complex. This is the entire model. And if you look at the equation, there's really nothing very complicated here. It's very easy to, and it's very close to the model I showed you. You just need to put a bit more non-linearity. It's not that difficult. Okay, so that works. We can model the data. And now, okay, so now we can actually look, given the model that was not fitted on most recent data, with using the robot, what we realized then is that so now we have all, and we also like many more ligand receptor compounds, and we can actually look at what happens for a library of actually here mutated cancer neoantigens. And then what we realize is that actually, if you look at this library of neoantigen, what you realize is that actually many ligands are antagonists. So that was a bit of a surprise because that sounds bad. If many ligands are antagonists, it means that if you have an immune response, it's very easy to kill it, basically. So that's not good news, but it is what it is. But then that gave us some ideas to actually monitor what was happening in immunotherapies, because if all ligands are antagonism, maybe you can have trouble when you use T cells to try to heal yourself. And so that's what led us to look at CAR-T immunotherapies. So I don't know how much you heard about CAR-T during this week, but CAR-T, the idea is that you take a T cell in your body, and then you're going to engineer the expression of a chimeric antigen receptor at the surface of the T cell. And the idea is that if you choose it carefully, the chimeric antigen receptor might be able to essentially recognize, attack, and destroy the tumor cell. So that's the idea. And then I've told you now, okay, you can have antagonism in the thing, and also you take T cells. So those T cells actually have endogenous TCR here. And so now you might be interested in seeing, could there be an interaction between the TCR and the CAR? And again, those are two receptors now. So we know if there is antagonism, it's not due to competition. It's really like, there could be interaction there, and there could be negative interaction, then you could have antagonism. And the bad news is that there is actually quite a lot of antagonism. And so we showed that. So that was done in collaboration with Naomi Taylor's group at the NIH. And so I'm going to show you 2 lines of data showing antagonism. So those are mice who have leukemia, induced leukemia, okay? And then we treat them with a classical CAR treatment. And so we look at the survival curve, and so they die, but they die roughly after 50 days. Okay. So now what you can do is that you can also get the cells, the leukemic cells, to actually express a strong TCR agonist. So now we have them expressing not only the CAR agonist, but also the agonist of the T cell that were used to do the CAR. And so, and that's the red line. And so then what you expect here, you expect it's kind of not a surprise, because now you have 2 ways for the T cells to recognize the leukemia cells. There is a CAR agonist, but there is also the TCR agonist. And so that's what you see. You see improved treatment, the CAR T cells, they destroy the leukemia cells. So that's good. But now the surprise is that if we put now a weak TCR agonist at the surface of the leukemia cells. So naively, you would expect that would not change nothing, because you will recognize mostly the cell will recognize the CAR agonist. But actually, what you see, what happens, you get a huge loss of survival. So the fact that you add a weak TCR agonist at the surface of the cell that are destroyed gives you essentially kill the immunotherapy. So that's antagonism in immunotherapy. And we also check that for what we call melanoma cells, so they call that solid cancer. We did the exact same experiment where you express at the surface of the tumor cells also either a strong TCR agonist or a weak TCR agonist. Remember, this is agonist of the TCR that is at the surface of the CAR. It's not the CAR, it's really the other receptors, the TCR. And then what you see is tumor size. And you see, so this is like normal immunotherapy. If you stimulate with a strong TCR agonist, the tumor are destroyed. But if now you also have a weak TCR agonist, you see that the tumor grows faster. So you basically neutralize the immunotherapy. So that's bad. That's bad. You are killing the immunotherapy. That's bad news. But those things can happen in your body because I've just showed you there are actually many antagonists in your body. So that's very bad. Okay. We can model that. We adapted the model. I don't want to spend too much time on the model because it's just to tell you we can model that, but we could really like look at quantify antagonism in all kinds of different contexts, and we feed part of the data. We can predict other part of the data. We did that properly with MCMC, blah blah blah. I want to, and that was an important step of the model of the process. Like that was done by my student François. It was super important to do to check what was happening and to see that we can actually really explain what was happening. But I want to spend too much time on it because I'm kind of running out of time. But I just have enough time to finish at 10:30. Enough slides to finish at 10:30. Okay, no, but I'm almost done. So. Okay, but now we saw that actually, so this antagonism thing is bad, because you're going to antagonize CAR T immunotherapy, but then there's actually a context in which it can be good. Okay, yeah, okay, that's kind of like a qualitative thing, but okay, I'm going to move to the context in which it can be good. Okay, so it's known in CAR T immunotherapy that you can have what they call off-tumor toxicity. So the off-tumor toxicity is the fact that, let's say you develop a CAR T to recognize a tumor, say a tumor in the ovarian cancer. So they did, the clinical trial, and then what they saw is that the cell, the CAR T were destroying the tumor, but they were also attacking the lungs of the patient. And then you can kill the patient because you have this kind of off-tumor toxicity that it's really, it was very difficult to do something very specific to the tumor. You tend to attack other part of the tissue. Okay, and that kind of makes sense because also I told you to some extent, you saw on my graph where we were trying to predict antagonists. We saw that mutation move you all around, so the self and not self are not that different. So that kind of makes sense. Okay, but then we realized that okay, maybe we can use antagonism to protect the tissue. Because we have also the TCR, and so the TCR could help us maybe modulate the response of the CAR receptor. And so we did that with the help of the model, and so it's just kind of like concept graph to show you what we did. So the idea is that now we have our CAR T cell, and so the CAR T cell has both a TCR and a CAR, and so we can engineer a CAR T cell so that the CAR recognizes the tumor, and also the TCR might also recognize a neoantigen at the surface of the tumor, but we can actually choose, we have freedom to choose this TCR. And it turns out that we could try to choose a TCR so that in the tissue that will be destroyed by the CAR, we can engineer it so that the self-antigen at the surface of the tissue slightly antagonize the CAR. And this way we can protect the tissue that would have been attacked by the CAR. So in the tumor, it's normal, nothing is really much changed. But the important thing is that in the tissue, the TCR that is weakly antagonizing the CAR can actually save the healthy tissue. So that's the idea, that's the concept. And so now our collaborators at the NIH and okay, so, and then, you have to choose this carefully, but this is possible. I don't want to spend too much time on that, but you can do that. Okay, so our collaborator at the NIH did this, we did that, and did the experiment. So this is a very cool experiment. You take a mouse and you inject the mouse both with an ovarian tumor and a human, actually, why is it lung? That's weird. Okay, that's a lung tumor. I thought it was ovarian. Well, anyway, that's a tumor and healthy tissue. So you have healthy tissue and you have a tumor, and then you have, you inject them with a CAR, different CARs done. So you inject them with just something that will recognize the tumor, the TCR, normal CAR, and then what we call AEBS CAR that is designed to antagonize. And then what we can show, we can measure the tumor, and they made the normal tissue luminescence so that you can really monitor in the mouse if the lung, a normal human lung, is destroyed or not. Okay, so those are the data, and you can carefully look at it, and you have to, it's not, I could do that, but what is interesting, this part of the graph summarize what is happening. So we look after 17 days. What is how is the healthy tissue, how it is changing, and how the tumor area, what is the tumor area in different contexts. So mock, nothing happens. You have healthy tissue is fine, and you have a tumor, so you're dead. TCR is just TCR, same thing. Now the normal CAR. So normal CAR, this is this off-target thing I was telling you about. So you destroy, so this is here, you destroy the tumor, or you prevent it to grow, but the tissue, healthy tissue, is destroyed. This is the toxicity of the CAR. And now with our AEBS setup, now this thing is rescued because of the presence of this TCR here. You can have at the same time the healthy tissue that is still okay, but the tumor area it is very low. So it's kind of the optimum. You've destroyed specifically the tumor, but you have not destroyed the lung tissue. So that works. And to do that, I should have insisted more on this because it's a math audience and not a biology audience, but you kind of need the model to somehow tune exactly the strength of the TCR to know where you are, to be sure that you are antagonizing the CAR. So that's where the model was useful. And we have kind of, there are rule of thumbs to do that, but we can really quantify that very precisely, and we know what to do to design this. Okay, good. I'm actually done. I'm going to wrap up. So I hope I convince you a bit, this kind of like all evolution, all travel from really like model explaining, this sensitive specific immune recognition based on quality that led us to do also this cytokine description and do a bit of machine learning to really understand qualitatively what was happening, extract this antigen quality dependency. And so we get something. Okay, I did not tell you too much about the interpretable model, but we could actually test it as well. And then now we're really moving to immunotherapy and how to use antagonism. We have studied a lot of antagonism and now in the context of immunotherapy. So the theory is published, the robot is published, this one is submitted. So we hope that things are going to work. And then I need to thank people because that was a huge amount of work over more than a decade. So Grégoire is my experimental collaborator. And friend. We've been working on this together for more than 10 years. The first adaptive KT proofing model was done by Jean-Benoît Lalanne, who was my grad student, master student at that time. And then all the recent work with the robot and CAR-T immunotherapy was done by François, Thomas, and Suraj on the experimental side. And the immunotherapy was done in collaboration with Nami Taylor and Taisuke Kondo. And I'm good. Thank you so much for listening.
I'm going to exploit the fact that I have the microphone to ask the first question. Thank you so much, Paul. I think this is such great work, and especially the last bit. And one of the things that I asked François, but I'm interested to also hear your thoughts on, does this maybe motivate a switch from CAR-T to CAR-NK? Obviously, the last bit you show that you can engineer these to delete that cell. But I wonder if having a more non-specific type of cell and CAR-NK, in pediatric leukemia, they're thinking about this or they're developing them already. So what are your thoughts there?
Well, I don't know much about CAR-NK, so I'm not sure I can really. The problem is really that. Are you sure the CAR-NK will not give you off-tumor toxicity?
No, so it could, but I think it's I'm not sure to what extent. But I just wonder if it's. The idea is that you have interferon gamma regulated.
I see.
The TCR and the NK receptors act differently.
No, that's a good idea, because you could imagine, especially if you have other, ways to control and maybe to, make it more locally specific. That could be a good idea. But I really don't know. I'm actually interested in learning more about CAR-NK because I actually don't know much about that. it's a good idea. We wanted to go more towards NK, like we want to put NK cells in the robot as well, just to see exactly what is the shape of the.
Okay, great. Well, let's talk. Because
Okay,
I'd love to hear about that.
that's on our to-do list. So that's interesting. You mentioned that. Yes.
Yeah, thank you, Paul. So I have two questions, one biological and one mathematical.
Okay.
So the bio question is your analysis useful to look at autoimmune diseases? So maybe there's an antagonist missing, which should be there or something.
Yeah, I mean, okay, so that's something I would love to do, but they work in a cancer institute. but definitely, so for instance, okay, I mean, related to that, so we have mice with, so Pavlov group at the NIH, they have mice that are essentially lacking the negative feedback. And so because of that, they have tons of autoimmune disease. So we see that. And we know it's related to the negative feedback, but they came to us because I was a bit surprised because they were basically expecting those cells to be like super strong and attack, work very well to destroy, but basically they destroy everything. So they were like, they did not understand why they lost specificity essentially, and that's because they don't have the negative feedback.
Interesting. Now, if I look at this diagram and tau and L, which you said, to me, it looks like a bifurcation. So can you identify a bifurcation in your ODE system, which would explain the transition?
Okay, I'm not sure. I don't think there's a bifurcation. So what do you mean by it looks like a bifurcation?
Well, okay, you have a TC, a critical value on the left.
Oh, I see. Right, so could this be a bifurcation? Okay, I see. I got it. Okay, so, okay. Okay, that's a great question because we suspect there is a bistable switch downstream there. So we suspect this is more like the control parameter for bifurcation indeed. But you still need, the way we think about it is that, this CN thing, I erased it, will be the control parameter of this bistable thing, but we still need this to define this control parameter. But there could well be indeed a bifurcation happening here. I agree with that. And we see, okay, so now we see evidence of that in the data. Like when we look at the data carefully, now we're looking at single cell markers, and we see evidence of an actual bifurcation and bistability. So we're exploring this right now, actually.
I'm an experimental immunologist, but okay. I'm quite curious about one of the slides you have. You plot the human and mouse T cells. It's something in the middle.
Okay, so I see. I think that was here. Yeah, that was the next one. That was this one, no? This?
Before that, yeah, there's a linear graph for.
Oh yeah. I see. Oh yeah, with the yeah. Okay. You see, yeah. This one, correct?
Yes.
Yeah.
So it's very interesting because the what we observe from the human and mouse T cell, they behave quite different, but based on your experience, it look quite similar. So could you comment on that?
Yeah, I can. For some reason, I cannot put it back full screen. I don't know. Okay, so why are they identical? You mean like why is there? Okay, I don't know. It's weird. Okay, so okay, that was a bit surprising to us first because we did not expect that to work so well. So what that really means is that, so for instance, I don't know if quantitatively the V0 here match. I did not check actually. I suspect they don't. Like the V0 EC50 relation is probably different in mouse versus human. But what is really important is more like the correlation. So the slope could be different. But what is really important is that the pattern that we see is the same. It correlates in the same way. But the values of the slope and all of that might be different. I never really look carefully at the difference. We were just like looking at the at the correlations, but
so what we observe is the first stimulation seems the same, but the second and third stimulation somehow the human T cell just lasts longer. And you can say because the human live longer, so T cell just die. So do you have some re-stimulation?
So okay, I will need to look carefully at the latent space, but it's well possible that you see that in the data, because this is just quantifying the correlation of parameters with one another, but it's not quantifying the actual values of the parameter. So it's perfectly possible that the T cells in human, they just live longer and that the time scale is different. That's perfectly possible, but I look carefully. I mean, I don't remember. I looked at them a long time ago, but we could have a look together, and we should see that. I mean, it should work.
Yeah, thank you.
Okay.
Thank you again for the lovely talk. I was wondering, in the estimation of these, well, I guess you can always well estimate the difference between N and M.
Yeah.
Do you see some kind of universality, or is very variable the number of these proof readings? Because I guess
no. Okay, so
is there some way in which is optimized also from that?
That's a great question. So as you can see from that, there are many models working. You know, you could vary N and M, and there will be like many antagonistic models. I mean, and that's why we had to change the model, even though the previous model was explaining part of the data. We had to change it because it was not, you know, when we had more data, we realized it did not work, and so we have to change the model. But it's more like a family of models. Okay, so based on what we do, okay, we can explain quite well the data with n minus m equals 2. So I think we take typically n equals 5 and m equals 3 or something like this. Okay,
and that use the same one across.
Yeah. But then, you know, the way to compute n and m, I mean, like if you look carefully, so you can get a better sense, and if you had better data, I think we could really like make very relatively precise prediction. Okay, so if I go back to, so now my thing is not playing. It's too bad. Okay, so it just moved. Okay. So n and m constrains very much the shape of these curves. So for instance, m is related to the slope here. And so the fact that it's flat here, it means that m is higher than 1. So you can do stuff like that. But again, this model is too simple. So we needed to update it, but there are things like that that you can do. And what I'm trying to do those days, I'm also interested in machine learning. I mean, I would like to have a systematic way to test these things, because I mean, we have a huge family of models that can explain the data, but I don't know how to explore this family of models. And so, and we're trying to do stuff along those lines, because that's precisely the problem we have. You know, we have all those data, we can generate many data, you know, data set like this, but you know, we expect that when we generate a new data set, our model is going to fail, you know, as we always expect. So we need a better way to build a model. We have to build this a bit by hand. With some intuition, but I would like to have a better way to do that. So yeah. Yes.
Hi, I was wondering if you could go back to the slide that had the weak and the strong, the TCR with the leukemia and the melanoma.
Oh yeah. That's this.
And I was just wondering if you had like a explanation for why the growth rate is faster with this.
Oh yeah, I see. Why the growth rate is faster? Okay. Yeah. Here, you mean why it's faster? Well, okay. We interpret it as antagonism. So how would we? So why is it a growth rate and not, say, just a quantity or something like that? That's a good question. I actually, okay, so I don't really know, but I could speculate on something we're doing in parallel that could, so we suspect, okay, so are you familiar with discontinuity theory of immunology? Have you heard about that? So there are people who think that a part of the difference between self and not self is essentially the growth rate of whatever is happening. So if you have a pathogen that is growing faster, it will induce a stronger immune response in general. And then part of the immune recognition is based on this. So I'm actually looking at this kind of models, right? I mean, we develop a model of discontinuity theory, and we also see antagonism there, okay, in discontinuity theory model. And so maybe something like this is happening, where you could have, growth rate sensitivity, and that could explain maybe a difference in growth rate. But I'm very much speculating here. I mean, we have to write the paper to explain that, but it's going to be a pure theory paper. But that could be something that could happen, and I would love to see. I mean, this is very much work in progress. I mean, I've been talking with my collaborators the other day about ideas like this, modulation of growth rate. Yeah, I have to say though that what is really crazy, I don't have the curve here, I think. But one thing that is really remarkable is that maybe I have the curve. No, I don't have it. But one thing I can tell you though is we can really. So if I go back here. We can predict, we can basically, the model predicts very well the intensity of the immune response, and it's measured, and this is done in the robot, so we measure the cytokine time course over, say, 3 days, okay, and the kind of antigenicity, the V0, the parameter of antigenic strength that we can infer for the model correlates extremely well with the eventual survival rate of the mice. So that means that after, we can measure the cytokine time course over 3 days, and we can predict if the mouse is going to live 20 days or 90 days. We can do that. And with the model, with this very simple model like this. So it's not clear what happens because, we're really looking at, completely different time scales, but that works very well. So we don't understand why, but I'm just telling you that works. So that's something I would love to explore more. Okay. Yes, Morgan.
Oh, no, sorry.
Okay.
I don't know why I raised my hand. Are there any other questions for Paul? If not, thank you so much for a wonderful last talk.